It's like the days go by slow, but the weeks go by fast.
Yeah, the days feel like 3 days, but the week is like,
It goes,
yep.
I'm like, I woke up and it's like, oh my gosh, it's Thursday.
But it's also been always a long weekend.
Exactly. I don't think it's like that.
I don't think it's been a long either.
Well, I didn't get the memo.
I bet you Verity started it.
Verity. He is, Allison did a good job on this, really bad.
There's Verity. Is that real?
No.
No,
Is it
you actually
real?
because actually you assaulted my family on you.
I'm sorry, I probably did. No, what's the really long word that you, I always,
So, Max and shake the anything and that shake is really shake again.
I tried to say that, but it
Yeah,
wasn't.
okay. What does that mean, like?
Oh,
Okay.
it's like along the lines of for your inability to desecrate something.
So you have one word for that?
Yeah, and you know how many times I have used? Every time I have to say the longest word is on game, and that's the only context I ever use this word in.
How many letters is it? Too many?
Yeah.
How many letters is it? Is it 30-some?
I don't remember. I have learned out now. One word. It's one word. I apologize for the question. I don't remember how it is. This is my own count. Like I could write it down and count it, but that would take a while.
Can you write out the word?
I mean, I could if I was okay with it.
Maybe after class.
Okay.
Does it take like extra minutes or what?
No. We have some long words too. German has some very long words.
I thought neuropsychopharmacology was a long word.
That is a long word.
Well, because the thing with Hungarian is we put like little slurs next to verbs. So the original word in what I just said is sang, which is sing. That's the word that we started. And then I put like little slots that change the meaning. What Germans do is they combine words. They don't really have slots that they put next to words, or not that many.
We didn't start yet, John, you're good.
Okay.
We'll wait till you sit down. It's fun seeing like a face up on the screen. You hear us, Alice?
Yes.
Oh, you're muted. We don't hear you.
Can you hear me now?
No, cannot hear you.
I'm
Oh, I
speaking.
guess you don't get to ask questions today.
I guess I didn't know you've done that.
Oh, wait a minute. Wait a minute. Okay, try it now.
Can you hear me now?
Oh, wait,
What about
maybe.
the owl?
The owl. Let me turn the owl up. Wait a minute. Okay, try it now.
Okay, can you hear me?
Yes.
Okay.
I like that noise.
Okay, then the Father, the Son, the Holy Spirit. Amen. God, grant me the serenity to accept the things I cannot change, courage to change the things I can, wisdom to know the difference. Father, the Son, the Holy Spirit. Amen. So before we start plugging and chugging with the neurotransmitters, let's just take a moment. Let's just review a little bit. So, what are some of the ways that we actually have been talking about What are some ways that drugs can potentially increase or decrease the effects of neurotransmitters? Okay, Bertie, how about give me?
One example would be the use of the receptors.
So you can what?
In the neurotransmitters that binds the alpha receptor.
Okay, so we can increase or decrease the activity of autoreceptors, right?
Yep.
Good. Okay. Okay, Julie?
They could bind to the receptor site so that the neurotransmitter can't bind.
Okay, so what do we call that?
Inhibition.
Inhibition? That's right. So we're going to go a little bit more technical today, or from this point forward, so we can inhibit, you said the receptor site?
Yes. Unless that's wrong, then I did not.
No, that's right, that's right. We call it antagonism. Good. That's blocking. We're blocking the receptor. We can block autoreceptors too. We can antagonize autoreceptors, or we can activate autoreceptors. Okay, good. What's another way? Devin.
Action potential propagation.
Okay, so we could increase, all right, so we could alter action potential generation in the neuron, right? Good. The neuron that releases that releases the neurotransmitter. Okay, that's good. We can certainly do that. Okay? Good, good. How about either Emily or Jonna, or Allison out there in
Would one of the probability of neurotransmitter release?
how could we change that with the drug?
Whether or not the action potential's received.
Okay, we did that. Devin sort of talked a little bit about that. So we talked about a couple other things. What are some other ways that we can alter neurotransmitter? How about altering neurotransmitter levels in the synapse? Let's get to that one. Altering neurotransmitter levels in the synapse. Looking at this row right here. This is the row I want to hear from.
I have no idea. Would it be like a reuptake inhibitor?
Boom. Good. Reuptake inhibitor. Good. What would you do? Okay, what would you do to reuptake? You'd inhibit it. Good. So you give a reuptake inhibitor. Good. We said that increases the neurotransmitter. Excellent. Julie, Allison, what's a couple other ways? Julie, Allison.
This could be just like a guess, and this could just not be true. Like the activation of enzymes, does that have anything to do with it?
Okay, activate enzymes. What kind of enzymes? What would these enzymes be responsible for doing? Go ahead.
I was gonna say the G protein receptors.
Those would be receptors for the neurotransmitter. So we could activate those with a drug, or we could block those. We've talked about that. Where are their enzymes? What do the enzymes in the synapse do? What do they do? What do they do? Julie?
They break stuff down.
They break it down. So typically, we don't activate those enzymes, but we inhibit those enzymes, right? We inhibit the enzymes that degrade the neurotransmitter. And this also leads to an increase in the neurotransmitter. Okay, good. So we talked about a lot of these. Just be sort of thinking about that. Whoops. Okay, and as we go through, as we're going through some of these things, if we're seeing receptors, we're seeing neurotransmitters being released, you might think, oh, I wonder how an inhibitor of the enzyme might affect behavior, or how antagonizing or activating these receptors might impact behavior, okay? All right. Because once again, ultimately, that's the goal, but we're still taking baby steps right now. So, first major group of neurotransmitters we want to talk about, catecholamines. They have this two basic molecular components, structural components, this catechol, and then an amine. We talked about its synthesis. So what are the two major catecholamines?
Norepinephrine and dopamine.
Good. So neurons, all neurons that synthesize catecholamines are going to have the enzyme tyrosine hydroxylase and this AADC. So dopamine is the first catecholamine that's synthesized. So dopamine neurons will have these two enzymes, but they're not going to have dopamine beta hydroxylase. They're just not going to have it. There are exceptions. You might have seen those in the book. There are some exceptions, but for the most part, dopamine neurons, they only make dopamine, so they can only take the process so far. Neurons that release norepinephrine, they also have this enzyme, dopamine beta hydroxylase, so they can do the final step. Add a hydroxyl group here. Or here. Rate limiting step, right here, tyrosine to dopa by means of tyrosine hydroxylase. So we said if I block or if I knock out tyrosine hydroxylase, I eliminate all catecholamines in the animal. They are lethal. They die very shortly after birth. And then if I block only dopamine beta hydroxylase, I'll still be able to make dopamine, but not norepinephrine. So I'll be able to enjoy my candy bar, but I might not be awake long enough to enjoy it. Yo! Okay, anyways. Now let's talk a little bit about dopamine. This is where we ended up, right? We ended up here on Tuesday. We've talked about these multiple times. So, since we have brought these two pathways up now 3 or 4 different times, they're pretty important, right? Especially for next Thursday. So 2 major neuronal, Julie?
The exam is Thursday.
I think so.
I thought it was Tuesday. Thursday's fine with me.
I mean, yeah, that would be wonderful too.
I'll look at the syllabus, but I'll go look at that. Oh, anyways, I'll look at it at the end of the class. All right. Yeah, Thursday's better for me too, because we can finish talking about the major neurotransmitters. But anyways, is it okay with you two? How about you, Devin? Next Thursday? Allison, is next Thursday okay for you instead of Tuesday?
For the exam? Yes, that's fine with me.
Okay, let's do that. Sweet. All right, cool. So substantia nigra. Allison's blocking the way. I'm going to move you. You ready?
Yes.
Be an earthquake. Where do you live? Did you feel that?
Yes.
So we talked about how these are, here are the neurons, these are the cell bodies of neurons in the substantia nigra. There's a control subject on the left, person with Parkinson's. Significantly reduced staining for dopamine neurons. So they begin in, this pathway begins in the substantia nigra. Nigra, striatal pathway. So we talked about that. It extends up to this region up here, which is called the striatum. What's one of the main functions of this pathway? Main functions. You see nigrostriatal pathway, Parkinson's, think movement. Good. Especially helps with smooth movements.
So you said the like decreased stain of dopamine, translates to Parkinson's. Would that increase dopamine? That's when we see symptoms of schizophrenia, right? Or would that work differently?
No. So that's correct. Yes. It's one of the major drugs to treat at least some of the symptoms, some of the positive symptoms of one of the oldest drugs used to treat some of the positive symptoms of schizophrenia is haloperidol, which is an antagonist of dopamine receptors. But one of the side effects is when you block them when you block the dopamine, the D2 class of receptors too much, you get some very characteristic movements like the tongue moving back and forth, pill rolling of the hands, they call it pill rolling, it's shaking. Very similar to what we see with Parkinson's. Okay.
Yep, so dopamine is necessary for muscle movement, like coordination of muscle movement. So dopamine affects basal ganglia in some way.
Yes, absolutely. This pathway it interfaces with the striatum, which is part of the basal ganglia. Good. You're pulling them together. Awesome. Yes. So one of the things that we can tell then is that, especially once we start talking about main drug effects and behaviors, if we start seeing an animal you give them a drug and you're just running all over the place, very likely it's going to be affecting this pathway in some way, especially in the striatum. Okay, good.
I have a quick question.
Yep.
For under ventral tegmental area where it says the second projects to the, what is the answer to that?
Okay. So the first pathway projects to limbic areas. So VTA, ventral, lower in the brain, tegmental, part of the mesencephalon, right? Midbrain, okay? The first projects to limbic areas, including limbic. How low can you go to the limbic? How low can you go? The limbic, no, limbo. Nucleus accumbens. We've talked about these areas a lot too. I keep hitting on them because we're definitely going to talk about these, I hope. The hippocampus, the amygdala, right? Because it originates in the midbrain, which is the mesencephalon, this is also called the what pathway. Mesolimbic pathway. And in general, again, we're just going to hit these again. In general, nucleus accumbens, that's rewarding, feeling good. Hippocampus, remembering that it's rewarding or feeling good. Amygdala, fear, danger. So all of these areas, all these brain areas are taking in these stimuli, and then when you activate this mesolimbic pathway, it has inputs to all those. Helps you consolidate and learn. This is something to be afraid of. This is something to go towards. Or when I'm stressed, I should go towards this. Okay. Second pathway extends to the prefrontal cortex. Otherwise known as the PFC, not KFC, right? And since the first pathway, the earlier pathway is called the mesolimbic, this is called the mesocortical pathway. We've definitely talked about this one a couple times. Mesocortical dopamine pathway. This is more cognition. Reduced, remember we talked about the dendritic spines. Remember talking about dendritic spines? And folks who are expressing reduced cognitive ability, one of the symptoms of that has been identified is a reduction in the number of the dendritic spines in the prefrontal cortex. So there's less ability to receive dopamine, perhaps. Okay, so those are the major neuronal pathways for dopamine neurons. Moving, feeling, learning. Dopamine's important. Okay. There are 5 subtypes of receptors, dopamine receptors. And they are simply, if you were to look at them or read literature about them, they would simply be called D1, D2, D3, D4, and D5. They are all G protein coupled receptors. So these all have that structural component, having that region that goes through the plasma membrane. When there's a site on the outside where the neurotransmitter binds, and there's going to be some G protein on the inside. That when that receptor is activated, this G protein is going to go into action and do whatever it does inside the cell. Okay. Functionally, these receptors are broken down into two major classes. We're going to call them subclasses, whatever, categories. I'm calling them categories. So the D1 and the D5 fall into, they are found postsynaptically. So what's that look like? Let's do this. My presynaptic neuron, here's my postsynaptic neuron, right? And I'm just going to call these in general, I'm going to just talk about D1. So when I'm talking about D1, it could be D1 or D5. You don't have to distinguish. Right? You don't have to distinguish that. So D1s are going to be over here. Postsynaptic neuron. Activation of D1 receptors leads to increased in the second messenger inside. So if I activate D1, so this is going to have an excitatory effect on the postsynaptic neuron. Okay. So they get dopamine release. The dopamine binds to and activates dopamine 1, these D1 receptors. There's my dopamine that comes in here, hits here. So DA, that's dopamine, plus D1, that's the receptor, equals that's excitatory. Right? D2. These are funky. So, erase this. Receptors that are considered D2, D3, or D4, these are all considered of the D2 category. And again, I'm not gonna hold you to this drug hits a D3 receptor. Well, we will talk a little bit about that, but I'm not gonna hold you to remembering that. The main thing is, well, D2s are found kind of all over the place. So I'm sorry to tell you this, but these D2 receptors, okay, first of all, they are found on the presynaptic neuron and the postsynaptic neuron. What? Okay. So if we drew our little synapse over here, we're gonna put some D2s here, put some D2s here. I'm gonna make them in squares. So that way we can see them. And I'm gonna draw them up here. Actually, outside of the synapse because they have a particular function here. So, if you activate the D2 receptors, you get a reduction in the second messenger. So in general, if I reduce the second messenger, is this inhibitory or excitatory? Good, it's inhibitory, good.
Did you say leads to reduction of the second messenger?
You got it. It goes, as they say in Pittsburgh, second messenger goes down. Get that?
Yes.
Understand that? You did, John? It goes down?
Yeah, down.
It goes down. Okay, it's gonna go down. So D8 plus D2, inhibitory. All right. So, that means that these things serve as autoreceptors and then I'm going to give you another effect that could happen. All right, this is just extra. This is for your information. Some of the receptors increase K efflux, which is also inhibitory. Okay, so that's more for your information. I'm not going to hold you to that. So these ones that are on the presynaptic neuron, we call those, here they are serving as presynaptic autoreceptors. Okay. Presynaptic autoreceptors. So, we get neurotransmission, we get dopamine, plus the D2 autoreceptor. What's the effect of that on the release of dopamine? What effect does that have on the release of dopamine? Where are they? They're presynaptic. So when I activate autoreceptors, what do I do to the release of neurotransmitter?
Inhibit.
Good. Yes. And if I activate the D2 on the postsynaptic neuron, what do I do to the postsynaptic neuron? If I activate D2 receptors, what happens to, I just in general, do I activate or do I excite or I inhibit the postsynaptic neuron? In general, I'm also inhibiting the postsynaptic neuron. That's correct. So by activating D2 receptors then, I can, or I do, depending on where they're expressed, either inhibit release, or it diminishes the effect of, or it inhibits the postsynaptic neuron.
And the reason for that is just simply because autoreceptors always have an inhibitory effect, right?
You got it. Yep. All right, so why is that? Does that seem confusing? If dopamine, dopamine is supposed to be the feel-good neurotransmitter. How does it ever exert its effects? Yes?
I'm guessing B2 and B1 receptors can't usually be found on the same neuron. Can they?
They are.
They are.
They are. So, what's happening? This is kind of complex, but we're going to try to wrap our brains around this today. So I'm going to represent action potentials in this neuron right here. So I'm going to represent action potentials traveling down the presynaptic neuron. Remember I talked about how they fire at different rates? So we said how they fire at different rates, right? Okay. Normally, this is the theory. On the left, well, first of all, if I were to vocalize this pattern, it would sound like bop, bop, bop. If I were to vocalize this one, it would be bop, 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 bop. All right? So first of all, which of these two would result in the release of more dopamine? Left or right?
The right?
The right. Oh, you can see that.
Yeah, a little bit.
Okay, so this is increased frequency, so I'm going to get more dopamine release here, less dopamine here. So the theory is that under normal circumstances, your dopamine neurons are undergoing what's called tonic firing, which means that those dopamine neurons, they're just sort of bop, 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 which means that they release low. Normally, your dopamine neurons are releasing very low levels of dopamine. So low levels of dopamine are normally released. And dopamine preferentially binds to D2. It preferentially binds to D2. So that means, under normal circumstances, maybe as you guys are sitting here right now, your dopamine neurons are just sort of firing dud, 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 dud. You're going to be releasing a little bit of dopamine, and most of that dopamine is going to be activating these inhibitory D2 receptors. Keeping things at a sort of chill out. When something stimulates, activates, whether it's something you need to move towards or good deal, whatever, something that you consider rewarding. This is something you get excited about. Ooh, I can't wait for that. Now those dopamine neurons are firing action potentials at a much higher frequency. They're releasing a lot more dopamine, and now all that dopamine is now hammering on these D1 receptors, resulting in excitation of this postsynaptic neuron. But in order to keep it from getting out of control, excess dopamine spills out. If those autoreceptors keep it chill, a little bit chill, all right? So under normal circumstances, you're probably, the dopamine that's released from your dopamine neurons are very low levels. It's those inhibitory neurons. Now, if there's something that's stimulating, something you find rewarding, something you find exciting, whatever feels good, you release a lot more dopamine, and you hit the D1 receptors.
I have a question.
Go ahead.
So you're saying that the lower frequency is inhibitory?
You got it.
And that's what we're seeing happening in D2 versus the higher frequency in D1.
Yep.
Okay.
Yep. That's why activation of D1. See that when you get this big rush of dopamine, that's one of the reasons why you sort of feel the difference. You can feel something being pleasurable because the dopamine releases it significantly increased, hits a different class of receptors, and it totally changes what happens in this postsynaptic neuron. In the nucleus accumbens, in the amygdala, in the hippocampus, and then those brain regions are connected with other regions which sort of pull a picture together. Good. Totally confused? Emily is.
So more dopamine is with the D1 and at the excitatory, and then low levels of DA with D2, and that's inhibitory.
That's correct.
Okay.
Normally we live in D2 lane. Yes.
That's what I was doing.
So normally, right? So this is or another way, maybe another way of thinking about it. Another way of thinking about it could be. Let's keep it on the rewarding side of things. Once you feel that something is rewarding or feels good, I bet you that that my mesolimbic, the dopamine neurons in my mesolimbic dopamine system are releasing a lot of dopamine right now and hitting those D1 receptors. That's why we can feel things that actually feel good. And one of the reasons that things feel bad is you could shut that down. Or people experience, we talked about anhedonia. Anhedonia, just nothing feels good. For whatever reason, these neurons, they just don't generate, they're just not excited. They don't become excited for various reasons.
I have another quick question.
Okay.
So for people that have depression, that would be taking an antidepressant, is that antidepressant then only going to be targeting their D1 category?
No. All kinds of neurotransmitters. Some hit norepinephrine. Some of them have some effects at dopamine, but the two neurotransmitter systems that seem to have more of an effect on depression is going to be norepinephrine and serotonin.
Okay.
They're all kind of involved, and I think by the time we finish with serotonin, by the time we get through dopamine, norepinephrine, and serotonin, you're going to say, oh, these things all kind of work the same. It's like a little stew going on in your brain. So what happens, Allison, is the other neurons can actually activate the dopamine neurons too.
Okay.
Right? So you can have inputs from the locus ceruleus that then activate, for example, that can activate the dopamine neurons. They're connected, right? But that's beyond the scope of this class. So let's talk about just a couple of drugs. Just a couple of drugs, right? General effects, very simple effects. Oh, look at a little mouse, a little rat, a little baby rat pup. Isn't that cute? 10 days old. So cute. Anyways, activation of dopamine, D1 receptors by this drug called SKF, developed by the, so drugs. Sometimes we're going to talk about drugs with a name, like quinipirole. Sometimes we're going to talk about drugs that have letters and numbers associated with them. SKF, that's the company that made it, SmithKline French. 393, that's just the drug number. So that's what that is. That's another name for a drug. SKF 38393. What do you think of general effect? General effect of activating dopamine receptor, D1 receptors with this drug would be? Okay, you could feel good. How about something that's more easily observed in an animal, perhaps? Yeah, increased locomotor activity. So we can just go right in and give a drug that activates D1 receptors. I think in general, which is very strange, is that in general, drugs that activate D1 receptors in general, if they're taken orally, they don't seem to have a lot of abuse potential. It's there, but it's not like some of the other drugs. It's very interesting.
So you feel like you get a combination with other drugs where they have more of the effect you're looking for with other
Is that
neurotransmitters?
professional? It seems as though part of it's due to the kinetics of the drug. For example, you could take L-DOPA, which is a drug used for Parkinson's disease. You take it, it's metabolized to ultimately ends up being metabolized to dopamine. And then that's not addictive at all because the rate at which dopamine levels are increasing in your brain taking L-DOPA, very slow, very low. So for drugs sometimes to really give you that, that rush effect, you need the drug levels to rise in your brain really quickly.
Okay.
That's another reason. Quinipirole, this is kind of a weird one, so I'm going to tell you right now, it's dose dependent. Low doses of quinipirole. But what do you think in general, what would happen if you gave a drug an agonist of T2 receptors? What do you think in general would happen to locomotor activity. Typically, it's going to decrease. But if you increase the dose, all of a sudden you get this shift, and you start seeing the animals will start grooming themselves and sniffing and sort of a lot of sort of atypical movement behaviors, which would kind of be similar to what we talked about with Parkinson's disease. And that's just due to the, once you start increasing the dose of drugs, this actually hits two different classes of dopamine receptors. So it's probably likely that as you increase the dose, you start hitting other receptors causing other kinds of effects. So, but anyways, you can end up doing that. And then finally, we've got overall, if you just give a drug, haloperidol, which just blocks dopamine receptors in general, you can get almost inhibition, lack of spontaneous movement. And what we see in this little rat, you little rat, is something that's called catalepsy. Catalepsy, you get lack of spontaneous movements, and this little animal, all right, at high enough doses, you place it, you give it the drug, you give it a higher dose of haloperidol, and then you place it. This is a very uncomfortable position for a rat, you can imagine. It doesn't want to sit there with its paws, front paws rested on this little platform and just sitting there. This thing had sat there for 3 minutes. No movement for 3 minutes. It's just sitting there. Totally out. That's catalepsy. Right, so just to give you some ideas of what can happen when we start dumping drugs into some of these systems. Locomotor activity is one of the things that will, especially with dopamine, that we look at. That's just a small little. Okay. Stop there for a minute. All right. Anything? Let's move on. Norepinephrine. Okay. The major neuronal pathway is this little group of cells that's called the locus coeruleus, and it's in the pons. So the pons originate. It's in the pons, part of the brain stem. And what's interesting is that the locus coeruleus is very small. It only contains around 50,000 neurons in the entire brain. So what I have here are some scans of some human brains. There's the locus coeruleus right there. There's little dots. There's my pons there, and here they are here. It's a little bit of a close-up. Here's the pons. And right there. Very small. Yet, locus coeruleus sends axons to almost every area in the forebrain. Almost every area in the forebrain. Including, like, the cortex, limbic system, a lot of the regions of the limbic system. We see hypothalamus there, hippocampus is there, septum is there. Also sends neurons to the thalamus, one of the sensory regions. So in the brain, the main source of norepinephrine are these neurons that are called the locus coeruleus. So you activate these, whoop, and the potential to activate the whole forebrain, and then go to the cerebellum, we're going to have some inputs for movement. So that's the brain. In the peripheral nervous system, neurons of the sympathetic nervous system release norepinephrine as a neurotransmitter onto target organs. Norepinephrine is also released from the adrenal medulla as a hormone. So when the animal is preparing for what kind of a physiological response to a stimulus, we're going to activate here.
Fight or flight.
Fight or flight. Fight or flight. And in the brain, we thought, up in the brain, we said that going way back to the beginning, one of the main effects, or one of the things that the locus coeruleus regulates is that, remember, arousal. Arousal, and paying attention. You got my attention? Yo! Right? Fight or flight. All you got to do is go out there and I could be kind of mean like that. I like to, if I see rabbits like nice and quietly chomping, I used to go like, when you do that, freezing, that's locus coeruleus right there, folks. Whoa. Then after a few minutes or 30 seconds, once you don't make any more noise and you're still, it's back moving. With us, we take that moment and we'd end up 3 weeks later, we're still like, what was that? Anyways, okay, that's locus coeruleus in general, big broad strokes. Julie.
Does norepinephrine ever work on like the amygdala?
Does it ever work in the amygdala?
I don't know like how, like,
Oh.
how to word that, because my thought is like the amygdala deals with fear, and oftentimes norepinephrine, like the fight or flight, relate that to fear, or am I just making stuff up?
So I think the way that works is that the amygdala sends, I think it sends neurons down to the locus coeruleus to get it like, you better do something and wake up.
Okay.
I think that's the other way. Okay, that's a great question. I think it goes the other way. And by the way, Julie asked a question about cyclic AMP and metabolism. I think she was confusing it with acetyl-CoA,
Yeah.
which is important for metabolism. Okay, you guys don't need to know that. So let's move on. Cool. That's a good question. I think that's what happens. The amygdala feeds down. Okay? Because I see something, I hear something, and sensory input. A lot of that sensory input, yeah, it's going to go down to the amygdala. The amygdala is the place where, yeah, that feeling, that's your amygdala is really jazzing out. Dr. Rhodes and spiders. Terrified of, sorry, Dr. Rhodes, I'm letting out your secret. freaks him out, bugs in general. Why is that? It hits his amygdala and then boom, down. Anyways, let's move on. Two major classes of receptors for this norepinephrine system, and while norepinephrine is one of the hormones that's released from the adrenal medulla, the more prominent hormone released from the adrenal medulla is epinephrine, which has another name of adrenaline. Oh, the Brits, they get to name everything. So, the receptors for epinephrine or norepinephrine are called, just like dopamine is dopaminergic, this is adrenergic. So these are adrenergic receptors. And they're all G protein coupled receptors. All right. Two major categories. We have alpha receptors. Why are they called alpha receptors? Alpha 1 was the first one discovered. Alpha, the first one they found. When they applied maybe adrenaline, or no, when they applied norepinephrine, boom, they saw this effect. Alpha 1. They are found on postsynaptic neurons, or tissues also, postsynaptic tissues, but we won't talk too much about that. Activation leads to, so this is alpha 1. Activation, so we've got norepinephrine plus alpha 1. That's going to activate. Remember, we're always talking about activating receptors, right? Leads to, ultimately, influx of calcium as a second messenger. So when you activate alpha 1, you get calcium ultimately. So activate alpha 1. Mobilize my G protein. Go G protein, go!
I think she missed it.
You missed it.
But it's recording, so.
Oh yeah, you get to see it all online. Woo! Anyway, so that ultimately is going to lead to opening of calcium channels. The calcium comes in. And in this case, calcium acts as a second messenger. Alpha 2. Oh, bing, bing, bing. Let's get to our, is this excitatory or inhibitory? Calcium coming in. Jonna. Boom. Good. EPSP. Good. Alpha 2.
Did you say excitatory?
Yes.
Okay.
Yes. By the way, today, DFD. What's that? Woo! Except for caffeine. Love it. Anyways. All right. So these are somatic. Nope, wait. They're terminal. The terminal autoreceptors. So they're here. These would be alpha 2s would be here. Okay. And there's somatic autoreceptors. Somatic, which means, that means that they're on the cell bodies. They're on the cell bodies of these adrenergic neurons. Don't worry too much about those. I want you to really focus on these. So we get norepinephrine plus alpha 2. Remember, we're going to activate this. Potassium efflux. Potassium efflux. So norepinephrine plus alpha 2, potassium efflux, that is inhibitory. All right, inhibitory. So we know how those two work now together. You inhibit, you activate the alpha 2s of the presynaptic, you're going to shut down release, right? We know how that works. What about beta? Beta 1, beta 2. Beta 1, beta 2.
So for the terminal autoreceptors, is that the same as the somatic autoreceptors as well, or is it different?
Somatic is going to act the same too. Yeah, it doesn't matter. The main ones I want you to. Ooh, I like that. That's a new chalk effect.
Oh,
I'm using lots of dopamine right now.
All these years of teaching, and you're still learning new tricks.
Love it. Okay. Beta receptors. You gotta entertain yourself because nobody else is gonna do that for you. All right. Beta receptors. Beta. So these ones were discovered second. So they discovered the alpha 1 first, then they discovered this other, structurally slightly different. All right, we got beta 1 and beta 2. All right, activation. Leads to increase in cyclic AMP, which is the second messenger.
What did you say?
And, yep, go ahead.
I didn't hear what you said. You said increased in.
Cyclic AMP, which means this is excitatory. And just by way of some information, in general, epinephrine is the one that, the hormone epinephrine is what interacts with beta 2 receptors. Okay. So this system is much simpler. Much simpler. Okay. Let's just hit a couple of, can I get that to leave? Here. Can I hide that? Why can't I hide that?
I think the arrow in the green, I think that, oh no, there's.
Yay!
Oh, it's silver.
Well, we're gonna flip it. All right. now I can't do anything. Okay, good. All right. Let's take a look at some drugs. We're going to look at wakefulness and arousal. Wakefulness and arousal. So I'm highlighting for you here a drug called phenylephrine. Phenylephrine, when you hear phenylephrine, does that even exist anymore, phenylephrine? Phenylephrine used to be very, very common in nasal sprays for congestion. For congestion. Notice the similarity in the structure. So here's our ring. See our amine on the 1, 2. It's on the second. It's in the same position. Then we have an extra methyl out here. Our hydroxyl is flipped. But otherwise, it looks very similar. So this drug, phenylephrine, binds to alpha-1 receptors. Now, the reason it works in your schnoz is that it activates alpha-1, which constricts blood vessels right in the nose, reduces secretions, decongest it. If you were to pop it, have some other effects. It's going to end up hitting your, could end up having heart effects. But, so I'm going to flip, let's see, I'm going to flip this green, take it out of here. Wait. All right. Okay, now we're going to let's look at the drug a little bit. Let's look at some data. Yay! So we're going to finally interpret some data here. So on the left, this is wakefulness. Here's our different treatment groups. By the way, bilateral means it's being infused into the hippocampus. So these drugs are being infused into the hippocampus in the brain. And this is phenylephrine on the left. This is isoproterenol, which activates beta-1 receptors in the middle. And then out here, we have the two of them separately, and then we have one infusion where they are both being given together. So what do you see happening? You guys take a moment and interpret those data. And by the way, vehicle, yellow line, that's like just whatever. Saline, whatever they're dissolving the drug in, that's what the vehicle is going to be. So the yellow line, consider that to be just no drug, but they're infusing something. So what are the effects of activation of these receptors, beta-1 or alpha-1 receptors on wakefulness? Alertness.
Do you mean just for that first graph, A?
All of them. All of them. So they gave the drug between pre-1 and post-1. They give the drug between pre-1 and post-1. They give the infusion between pre-1 and post-1.
What does that mean for one and next one?
It's just a time. So they had time. They were basically, so pre-2, at some time before they gave the infusion, I'm glad you asked that. They made observations about the animal. Is it awake? Is it not awake? How much is it awake? In a certain period of time. So pre-2, that's before, way before. Pre-1 would be somewhat before. Then they give the infusion, And then we're looking at different time periods after the infusions. Yep. And then this one is just post one time after a week, or one time a week after infusion. So this is more about, maybe just look at A and B. A and B is probably better to look at. So what effect did activating these receptors have on wakefulness? Did it increase it?
It looks like it's increasing arousal and wakefulness.
It looks like it's increasing arousal and wakefulness. What do you guys think?
I think it matters the different concentration of, you know.
Phenylephrine?
Yeah, the phenylephrine, because it says, the 40, it has like, it's longer and it's more of like a spike, it like goes up immediately, and then it kind of just like gradually goes downward, and like the lower levels, it takes longer for it to peak, and it doesn't like peak for as long as compared to like the higher.
Okay. Okay, good. Any other thoughts? So it's dose dependent, so they definitely are more awake after they've had the drug, right? They're more awake at least for a little while. Are they more awake with isoproterenol compared to the yellow line? Are these lines higher?
Yeah.
Okay. Which of those two has which of those two, if you wanted to wake somebody up, well, actually, if we look at the combination, here we see that they're both combined, we get somewhat of a, so both of them combined, they're awake more than each of them by themselves. But which of these probably is making the bigger contribution to the effect? Alpha 1 or beta 1, and why? If you're going to make a guess. Julie?
Beta 1 is acting as the excitatory way.
Looking at the data. So look at the data. If I activate alpha 1, this is alpha 1, this is beta 1. Which of those two actually?
Oh.
Okay. I'd say alpha 1 maybe. Here they look the same. But I think we can conclude they're both important. They're both important for wakefulness and arousal. So when your locus coeruleus is activated to get you to pay attention to something, or to be alert, it's likely that in your brain, both alpha 1 and beta 1 receptors are being activated. They're probably working together. Okay, so, whoa, hey, we reached the end of another day. Does this class, okay, does this class seem to go fast to you?
Yeah.
Yeah, it does.
Okay, because it's like, I looked down, it's like, holy cow, it's been an hour and 15 minutes already. Awesome. So, for next week, so the exam is Thursday. We are going to talk about, we're going to finish with norepinephrine. We're going to start talking about serotonin. We're going to hit acetylcholine, which is the best in it now. And then, I don't know if we're going to hit GABA, and we're probably going to hit, not going to hit GABA and glutamate. Awesome.
So what we're saying is, instead of coffee, go for the
nose spray.
I'm just kidding.
I don't think you should start charging those spray, that's deal.
Phenylephrine, you can't get it anymore? So,
That's just killing me.
How about the ISO?
you don't want isoproterenol. No, it's just not, it's actually,
Coffee is.
that is actually used more for
when